This is Bernard Paulson. We are going to discuss enzyme kinetics today. In our previous lecture, number 4, we talked about chemical reactions, whereas here we will talk about enzyme catalyzed reactions. The outline of this lecture is as follows. I will first speak to the principles of enzyme catalysis, where we talk a little bit about enzyme properties, their biochemistry, and how we think about them, and how they actually function biochemically. I will describe the general process that is used to derive rate laws for enzyme catalyzed reactions. That will be followed by detailed description of 3 important reaction mechanisms associated with enzymes. The first one is called the Michaelis-Menten reaction mechanism, and the kinetics that come with it are prevalent throughout the life sciences. We will then talk about the Hill kinetics that are used to describe regulatory enzymes, and then we will talk a little bit about the symmetry model that is an accurate representation of regulatory enzymes. Throughout this slide deck, I have comments about how one uses scaling of equations to deal with, to mathematically deal with enzymatic rate laws, but most of this material I will skip in this voice recording. However, the slides are found throughout this collection. So we will begin with some basic information about enzymes and enzyme catalysis. This slide here illustrates some of the basic concepts. Panel A shows with that yellow bar the turnover of a reaction without an enzyme, whereas the orange bar represents the turnover with an enzyme, and the blue is the difference between the two. It is frequently stated that enzymes accelerate chemical reactions, and those acceleration factors can be as much as 1,000,000-fold or even greater. I discussed some of the principles of how enzymes work through surface chemistry and bringing reactants into the right proximity to vastly increase the probability of a reaction taking place. The panel B on the right illustrates a couple of important concepts. So the first row of schematically represented compounds shows a squiggle bond and a straight bond between various chemical moieties. And these moieties are represented by squares, circles, triangles, and so forth. If an enzyme has high reaction specificity and high substrate specificity, it'll only act on one bond in a very particular place in a particular molecule, and this is represented by the yellow circle labeled A, where you have a squiggle type bond between a circle and an upward pointing triangle. Furthest to the right in the same row, we have a squiggle bond and 2 triangles pointing opposite to one another, representing a different compound. And if the enzyme does not act on that, it has a very high substrate specificity and only cleaves that bond, the squiggle bond, when it's between the circle and the triangle pointing upwards. The second row shows an enzyme that has a high reaction specificity but low substrate specificity. So it will cleave a bond B in green between different chemical moieties on a molecule. So something like aminotransferases, they tend to have low substrate specificity and are able to carry out all sorts of enzyme transaminations, as an example. And of course, example C is when an enzyme is highly promiscuous, as is called, with respect to substrate and what type of reaction it carries out. And some of the principles of enzyme catalysis are detailed in this slide. In panel A, to the top on the right, we see a reaction take place in solution between A and B. If they collide as reactants in a particular configuration where the right chemical groups line up, they may enter a transition state by overcoming an energy activation barrier. Covalent bonds are broken and formed, and you will get molecules C and D to be formed and released. Panel B shows how this happens on an enzyme, and there is a cycle that starts out with bullet point 1, where the enzyme is free. It has an active site. It'll first bind to reactant A, then to reactant B, and they are put into that perfect proximity in the active site of the enzyme. And under bullet point 4, you see that they enter a transition state by interacting with different chemical moieties on the enzyme molecule. That triggers the reaction, and then the green compound C is released, and then D, liberating the free enzyme that is now ready to undergo another cycle of catalysis. The enzyme commission number, or EC numbers, for enzymes classify what type of reactions they carry out. It is a 4-digit code as shown to the right of this slide. The first digit refers to the major classes of enzymes, and there are 6 of those, and they are illustrated in this slide. So the first class is an oxidoreductase that carries out the exchange of a redox equivalent between a blue and a pinkish molecule, as shown. Transferases take a chemical moiety, here shown in yellow, and transfer it between 2 other groups, A in blue and C in pink, as shown in the second row in this table. Hydrolyses are enzymes that break bonds, ester bonds, peptide bonds, other bonds, where a water reacts with a bond and breaks a compound into 2. A lyase in row 4 is an enzyme that takes 2 moieties, A and B, and glues them together. And there's the several families of those. An isomerase, as you note, has only one reactant and one product. And this is actually the same molecule in terms of chemical composition, but they're slightly rearranged. And we talked about the phosphoisomerase in the glycolysis in earlier lectures. That falls into this category. And then finally, there are the ligases that bind together 2 molecules, A and B in this case, and use a cofactor, often an energy-containing cofactor like ATP, to glue 2 molecules together. So these are the basic 6 types of reactions catalyzed by enzymes, and these 6 types are the basis for the first digit in the EC number. For specific enzymes, we can get a lot of information. Here's an example from the protein database. This is an oxidoreductase, specifically a malate dehydrogenase that is in the TCA cycle. Here is a depiction of the enzyme, a 3-dimensional structure. In this case, there are two subunits to the enzyme that are the same, and they form a homodimer, that is the active form, and that enzyme can catalyze the reaction shown below, where malate goes to oxaloacetate, and an electron is transferred to, I should say, a redox equivalent is transferred to NAD to form NADH. Notice the enzyme commission number. The first digit in this 4-digit number is 1, meaning it belongs to the class of oxidoreductases. This sort of information is available for a large number of enzymes in this database as well as others. So that gives us a little bit of an introduction to enzymes, what they do, how we think about them, and how we can get information about them. In terms of this class, we now need to figure out how to describe mathematically the action of enzymes when it comes to catalyzing reactions. The process that we go through mathematically in deriving enzymatic rate laws is as follows. First, we start with a postulated reaction mechanism on which we formulate dynamic mass balances on all the elementary reactions found in that reaction mechanism. And we'll go through examples of these. We then find the time invariants, or the mass balances, things that don't change with time. We then use those mass balances to eliminate dependent variables, to reduce the mathematical description to contain only the dynamically independent variables. The equations that describe that minimal set of dynamically independent variables are often mathematically intractable, meaning you can't solve them to get a closed form solution. To get around that, historically, simplifying assumptions were applied, with the two principal ones being the quasi-steady state assumption and the quasi-equilibrium assumption, and we apply them and derive simple equations to describe the reaction rates. In this day and age, it is very easy to numerically integrate the full set of equations as well as the simplified ones to determine where the assumptions apply. I should note that this step 4 was carried out for historical reasons. These assumptions were developed, were applied and used to develop rate laws in an era where we didn't have readily available computation, and we needed them to get mathematical solutions. In this day and age, we can simply numerically simulate the full set of solutions and never apply these simplifying assumptions. To look at the validity of assumptions and understanding what the rate laws say as equations, they are often scaled, made dimensionless, and we figure out what the key dimensionless groups are and what the right numerical values for them are relative to the applicability of certain kinetic assumptions. That's a lot of words, and we will not go through this topic here, as this is the subject of graduate level treatment of this material. So that concludes the introductory comments on enzymes and how enzymatic rate laws are derived. We will now proceed and apply this procedure that I just outlined to 3 important reaction mechanisms. The first one is the Michaelis-Menten reaction mechanism. It was postulated in 1913 with the now frequently used rate law named after the mechanism that I will now derive for you. The mechanism is simple. It assumes that the substrate binds to an enzyme reversibly to form a complex, and then the reaction takes place that comes with the release, the irreversible release of the product and the liberation of the free enzyme. For many enzymes, it turns out that the binding step is fast relative to the catalysis step that should be considered slow in comparison. Now, we can begin to apply the process outlined to this reaction mechanism. So first, we can write out the dynamic mass balances as shown to the left of this slide in the center set of boxes. The stoichiometric matrix has 3 columns that correspond to the 3 elementary reactions, and there are 4 rows in this matrix that correspond to the 4 compounds involved. We can multiply the stoichiometric matrix times the vector of fluxes to get the flux balances shown. The fluxes can be expressed as elementary rate laws because these are considered to be elementary reactions, and these are written in there. And for instance, the binding step is K1 times the concentration of substrate, S times the concentration of free enzyme E, and so forth. The rank of S is 2, which means there are 2 degrees of freedom here, dynamic degrees of freedom, but we have 4 concentrations, so there are 2 redundancies in this system. One of them is immediately apparent because we see the second and third row in the stoichiometric matrix that correspond to the enzyme, the free enzyme, and the enzyme complex add up to 0, and therefore the summation of E and ES is a time invariant as shown. The other time invariant is the substrate that is either free, bound to the complex, bound to the enzyme to form the complex ES, or been liberated as product. In the central panel to the right, we show these 2 time invariants, and if we are doing an experiment in the laboratory, we can fix the total amount of the enzyme as E0, the initial condition for starting the simulation, and the substrate at time 0 as well. That is the total amount of substrate in the system. The matrix L is just a representation of the 2 row vectors that correspond to these 2 conservation quantities. So the first row has 0 on S, 1 on E, 1 on ES, and 1 on P. And when you take the inner product of that row and the concentration vector, you get E plus ES. And similarly for the second row in L, you get the conservation quantity on the substrate. M minus R in this case is 4 minus 2, which means there are 2 time invariants, and they are shown by the 2 rows in the matrix L. The next thing we do is to take the mass balance, dynamic mass balance equations, substitute in the elementary rate laws as shown, and we have now 4 differential equations, but we can eliminate 2 of them by introducing the time invariants. And historically, the free enzyme concentration is eliminated from equation 1 and 3 by substituting E naught minus ES wherever E appears, and the product is eliminated by the conservation on the total substrate as shown at the bottom of the slide. This leaves us with 2 independent differential equations, dynamically independent differential equations for S and ES, namely the substrate and the intermediate complex. And these right-hand sides are described by two functions, f1 and f2, that are functions of the two dynamically independent variables. So if we choose S and ES, we now have a model of two differential equations that have two right-hand sides described by f1 and f2. And then we have two algebraic equations from which we can calculate the enzyme and the product when we know S and ES. And to do that, we have two functions, G1 and G2, which are the equations describing the time invariance. Now, it turns out that F1 and F2 are of a certain class of differential equations called Abel-type differential equations, class 1. And one can show that they cannot be solved in a closed form. So you cannot get solutions to these differential equations. They are bilinear, by the way, in their nature. So historically, to get around this problem, an assumption is applied, which is called the quasi-steady state assumption. And it assumes that the intermediate complex is in a steady state. And one can put the time derivative of it to 0. This means that the function f2 is equal to 0, and it can be rearranged so that we can calculate the concentration of the intermediate complex as a function of the substrate concentration that we call here f3. We then take the differential equation for the substrate f1, and we substitute in as the second argument f3 for ES, and now we have one differential equation in one variable, and can solve it, and as we solve it, we get the famous Michaelis-Menten equation. I see now that there's a slight problem here in the last equation with the argument list in the function f1 is simply supposed to be s1 comma f3 of s. This is the famous Michaelis-Menten equation that comes with a V max value, the maximum velocity of the equation, and the Km value in the denominator. We can plot this reaction rate law as a function of the substrate concentration, and that is this familiar hyperbolic rate law, where you start out with the reaction rate on the y-axis growing rapidly as a function of s, but eventually leveling off and reaching a maximum rate of v1. The half height of that function, this is called the hyperbolic function, is found at a substrate concentration equal to Km, and if you substitute Km equal to S into the Michaelis-Menten equation, you get that the reaction rate is half of V max. There are two limiting behaviors of this rate law. One is at very, very large substrate concentrations relative to Km. You can readily see that the rate law goes essentially to a constant, in that case, V max. And the other extreme is when the substrate concentration is very small, goes to 0. And of course, that means relative to Km. In that case, we have linear kinetics with k cat times S giving us the reaction rate. Interestingly enough, for most situations in vivo, the substrate concentration is close to the Km value. This means that the half of the enzyme capacity is used, and enzymes are expensive to make for a cell, as protein are. But also it means that the reaction rate is responsive to changes in the substrate concentration. So as usual, that seems to be an optimal condition satisfying these two constraints, or these two considerations. Now we can simulate here, put the equations on MATLAB or Mathematica and simulate, and we can simulate both the full set of equations, the two independent differential equations, and the simplified form, which is the single differential equations on the substrate. And calculations from such calculations are illustrated in this slide. I show the numerical values used in that box to the top, and this is a situation where E naught, the total amount of enzyme, is 100 times less than the Km value. The top plot is a phase plane of S versus ES, the complex, and we start with substrate and no complex. So we start at the point 1, 0 in that phase portrait. The dashed line shows the full solution that moves very rapidly vertically away from that initial condition towards that black line, that solid line, which is the quasi-steady state solution, and then moves along it into the origin. At time infinity, all the substrate has been converted to product because of the irreversible reaction in the reaction mechanism. The solid line is the quasi-steady state solution, and you can see that after that initial vertical motion, the two are essentially identical for these parameter values. The red box shows the rapid response for substrate and the complex. And we show here the dashed line, the full solution, and the solid line, the quasi-steady state solution. For the substrate, the two are virtually identical, not changing much on this fast timescale. The solutions for the complex are different. Of course, the complex starts at 0, builds rapidly up to a value, the quasi-steady state value, in approximately 2 time units in panel C, whereas the quasi-steady state solution assumes immediate establishment of the quasi-steady state, and at time 0 we are at that value. So the difference between the dashed and the solid curve in panel C is the error made by applying the quasi-steady state's assumption, and that is minor for this set of parameters. The green box shows the slower response over hundreds of time units, and we see the solid and the dashed curves are virtually identical. In panel E, you will see a dashed curve climbing very rapidly along the y-axis, and that rapid rise happens over the first time unit that is blown up in panel C. So these graphs show the dynamics of the Michaelis-Menten reaction mechanism, the full form of it and the simplified form of it. And in this particular case, for the numerical values chosen, the two are indistinguishable on this slower time scale, which we are mostly interested in. Here is another simulation for a different set of parameters. Just as a historical note, the conditions stated for the validity of the quasi-steady state assumption were that the initial enzyme concentration, E naught, had to be much smaller than S naught, the initial substrate concentration. Detailed analysis and supported by simulation, as shown in here, shows that the actual criteria is that E naught has to be much smaller than Km. So on this slide, we show a simulation where the initial conditions are such that E naught is equal to S naught, and both are much smaller than Km. So as before, the phase portrait shows a rapid rise in the trajectory away from an initial concentration of S at 0.01 towards a quasi-steady state line that is now essentially a straight line. And then it moves along the quasi-steady state solution into the origin. The red box shows the fast response, and again we show that in 2 or 3 time units here the full solution converges to the quasi-steady state solution, and in the green box we show that on the slower time scale the full and simplified solutions are essentially the same. This shows how simulation can easily check the validity of assumptions that lead to the use of simplified solutions. Historically, the literature would have told us that under these parameter values that this quasi-steady state assumption would not be good, but it actually is. Because in this day and age, we can rapidly check all these things quickly by simulation. Now here are just some results that show under which parameter conditions these assumptions apply, and there is one condition under which the quasi-equilibrium assumption applies, as shown in the first row or first line in this table. So these kinetics are known as Michaelis-Menten kinetics, and now you all know that the Michaelis-Menten equation is based on a quasi-steady state assumption that you really don't have to do any longer because you can simulate the full solution relatively easily, even if you had to treat a large number of enzymes simultaneously. The next 2 rate laws we are going to talk about apply to regulated enzymes. And let's start out here by looking at panel C that shows the R and T conformation of a multimeric enzyme. R stands for relaxed and normally refers to a conformation where the active site is open, whereas T stands for tight or taut, where the configuration of the enzyme or the conformation of the enzyme has been changed so that the active site closes off. So in this particular case, there are 6 catalytic subunits to this complex denoted in blue. Each one of them has an active site, and then there are 4 regulatory subunits in white, or I guess 3 in white, that tie the catalytic subunits together. On the regulatory subunits, there's a binding site, what's called an allosteric binding site, for a molecule that is different than the substrate or the products of this reaction. That molecule is called a regulator. It can inhibit or activate an enzyme. As you can see in the upper cartoon, when the when the green molecule is bound to these regulatory subunits, the enzyme is collapsed into a tight form, and the active centers or the active sites, denoted by pink, are closed off. In the lower cartoon, there is no green molecule bound, and the whole complex is relaxed, and the active sites are open on the blue catalytic subunits, and the enzyme is ready to catalyze a reaction. So there are many different ways to describe the reaction mechanisms associated with regulatory enzymes. We will discuss 2 of them: Hill kinetics and the symmetry model. Hill kinetics are frequently used, however, they are oversimplified in terms of the reaction rate mechanism and are unrealistic in terms of the underlying the underlying chemistry that takes place, whereas the symmetry model does represent a realistic situation for a large number of enzymes and enzyme complexes. Nevertheless, Hill kinetics, due to their simplicity and applicable assumptions, simplifying assumptions, are frequently used. So, the reaction mechanism for an enzyme following Hill kinetics is shown here. We have two chemical equations. The first one shows an enzyme binding to a substrate and in just one step converting into a product liberating the enzyme. The second equation shows the enzyme binding to a number of inhibitors denoted by I to convert it into a different conformational state denoted by X where it's inactive. And this is an equilibrating reaction. The simplifying, so there's the inhibitor I, there's the catalytic form, and then the quantity nu is the number that makes the Hill kinetic mechanism unrealistic. We all know that most reactions are bilinear. So if the assumption is that nu is 4, so let's say 4 inhibitors are known to bind to the enzyme, we put 4 in there and we assume that 4 inhibitors bind simultaneously to the enzyme. Of course, that does not happen, and this represents a lumped reaction. Lumped reactions were introduced in lecture number 2. However, you can fit this rate law that we are about to derive to experimental data and compute what that coefficient nu is. And for instance, in the case of oxygen binding to hemoglobin, the measured number is 2.3 to 2.6, whereas we know that 4 molecules of oxygen bind to hemoglobin. So nu can be a fractional number and therefore doesn't represent actual chemistry. Of course, hemoglobin is not an enzyme. It's just loaded up with oxygen, and Hill kinetics were originally developed for hemoglobin, oxygen binding to hemoglobin, but since then were adapted to enzymes where the bound state was catalytically inactive. There will be a mass balance here. The enzyme is either free or inhibited, and that's a conservation quantity. In deriving the rate law, we are going to make a quasi-equilibrium assumption on reaction number 2, and that just means the forward and the reverse reaction are equal and opposite, and that leads to a relationship between X and E as shown. And although there is not an actual number of binding sites, because nu is not an integer, the equation is normally written in the format given, where Ki is a per site, in quotation marks, binding constant. And of course, there is not an integral number of sites here. The reaction rate itself for reaction number 1 is a rate constant times the concentration of the substrate and the enzyme. And what we seek to do here is to eliminate the enzyme from this elementary rate law and replace it with the concentration of the inhibitor, so we can track out the reaction rate as a function of the concentration of the inhibitor. This slide shows how we actually derive the rate law. We start out with the mass balance and the quasi-equilibrium assumption, and we substitute the expression for X in the quasi-equilibrium under the quasi-equilibrium assumption into the mass balance. And this gives us a relationship between the free enzyme concentration and the inhibitor as shown. The parameters here are the total enzyme concentration, the per site binding constant, and the degree of cooperativity. We can then substitute this into the rate law to eliminate the concentration of the free enzyme and get the Hill rate law as shown. So you can see that the concentration of the inhibitor appears in the denominator. As that concentration grows up, I should say builds up, the denominator grows and the reaction rate goes down. You can go through the exact same derivation, but under the assumption that nu, when the nu molecules of A, the activator in this case, are bound to the enzyme E and forming X, that X, the bound state of the enzyme, is now catalytically active, not inactive, and it can catalyze the same reaction as E, but at a higher rate. In that case, you can derive the Hill rate law for activation as shown down there. I often put that on midterm exams. I see now, looking at this slide, the reaction shown here is incorrect. S plus X is converted to P plus X, not E, with a rate constant of K prime. In any event, this is the Hill rate law shown on the bottom left and for inhibition, and the Hill rate law for activation is shown on the bottom right. We can graphically represent these. Let's start with the inhibition. So we can plot the rate law as a function of I, the inhibition constant, and we get an S-shaped curve that drops towards 0, and it has an inflection point where the derivative is maximal, and this means that the rate is maximally sensitive to changes or fluctuations in the concentration of I. The activation form of the Hill rate law mirror image this. It goes up to a rate of alpha times B max, where that corresponds to everything being in the form of X and proceeding with the K prime rate, and saturates eventually at that level. The range of concentrations for the inhibitor or the activator have a maximum sensitivity where the rate changes the most. If it is too high, if their concentrations are too high, they really don't influence the rate law much. And if they're too low, there is little sensitivity as well. I should point out that these feedback mechanisms are often found in biosynthetic pathways to products like amino acids that are used for protein synthesis. So if the rate of protein synthesis goes down, the concentration of the amino acid builds up, and that feedback inhibits the first reaction in the pathway that leads to the amino acids, slowing down its rate of formation, and vice versa. If the protein synthesis increases, the concentration of the amino acid drops, and as it drops, it would alleviate the inhibition of the input into this pathway. So that is the way regulatory enzymes do their function. They are involved in feedback or feed-forward inhibition, as we shall detail in lecture 9 later on. Now, on to the subject matter of this course. We can simulate these equations. In this case, the full equations without the assumption of quasi-equilibrium. And that is what's shown in this set of diagrams. These are phase planes between S and E, E and X, and the inhibitor I and X. So the first phase plane shows an L-shaped curve from time 0 to time infinity. First, there is a vertical drop as the inhibitor binds to the enzymes to convert some of it into an inactive form. Then that reaches equilibrium, and then the active form catalyzes the reaction, leading to a horizontal motion associated with the depletion of S, while the free enzyme stays at about 80% of the total. The parameters used in this simulation are shown in the red box. The phase plane between X and E is a straight line with a 45-degree slope, negative 45. This corresponds to a mass balance on E and X. And we start with only E and no X, and we rapidly shoot up to that equilibrium point where 80% of E is in the active form and catalyzes the reaction. The last phase plane also has a straight line because the inhibitor is conserved in a closed system. It's either free or bound to the enzyme. And this little equation there shows the this conservation quantity, and it's reflected in this straight line in this phase plane. We can also look at the transient responses here on the fast and slow time scale. On the fast time scale, you see the substrate and the product haven't changed much, whereas the enzyme concentration, the inhibitor concentration, and the concentration of the inhibited form rapidly go into equilibrium state. And this happens in 1/100th of a time unit. And then if you look at the slow dynamics unfolding on a few time units, during that time, E, I, and X have flatlined, whereas the substrate is dropping and the product is building up. So that's how we would simulate and describe the dynamics of the Hill reaction mechanism under the parameter values given. So that's our second rate law treated in this lecture. The third one is the symmetry model, and this is a chemically realistic mechanism for inhibition. The derivation of a rate law here follows the same procedure as for the Hill rate law, except the mathematics get a little more cumbersome, and I will not detail them as thoroughly as I did for the Hill rate law. You can go through the procedure if you like, and it mirrors that for the Hill rate law. So first, we look at the reaction mechanism. This reaction mechanism relies on the observation that an enzyme can naturally exist in two forms. It forms an equilibrium between two conformational states. Here, they are denoted by E, which we take to be the relaxed form. And this is a tetramer shown, where all the subunits have a square conformation where the active site is open. The X form is the inactive form, where the protein subunits naturally take on another conformation in which the active site is closed off. The way an inhibitor works in this case is that it can bind to each one of the subunits, not in the active site, but in an alternative site, and it's called an allosteric site. And we have therefore four binding steps. First, an inhibitor will occupy one of the subunits, then to form an X1, then that X1 can bind to another inhibitor molecule to find an X2, and so forth, culminating in X4, where all the bound sites are occupied. And note that the rate constants for the forward and reverse reactions shown counts up each open site and each closed sites in these numbers 4, 3, 2, and 1, and so forth. So K plus and K minus are a per binding site rate constants. And if there are 4 of them open, you get 4 times that forward rate. The so-called allosteric constant L describes the equilibrium between the active form and the inactive form. So the more inhibitor is present, by simple mass action kinetics, more and more of the total enzyme is drawn into the inactive form. So it could be X1, X2, X3, or X4, or even X itself. So the summation of these 4 forms are all representing an inactive state of the protein. All right, now we can go on and derive the rate law. First, we look at the mass balance. The enzyme, the total amount of enzyme, is either free or in one of the inhibited forms as shown. So this is more complicated mass balance than I had for the Hill rate law, where I just had E and X. Here I have 4 additional inhibited forms of the protein. So there are a total of 6 states that this protein molecule can be in, and the summation of all of them is a constant equal to the total amount of enzyme present. Next, I do some mathematics. I have to make 4 quasi-equilibrium assumptions for each of the binding steps, and that's the column of equations shown to the left. And I can solve this in a nested fashion as shown to the right. I get x1, 2, 3, and 4 expressed as a function of x and I. I can then substitute all of these forms into the mass balance for x1, 2, 3, and 4 as shown. And then in the second line, I can combine these based on the Pascal triangle. These form a perfect fraction. And then I assume that E and X are also in equilibrium as given by the equilibrium constant L, the allosteric constant. And I can now eliminate X from the description, resulting in a relationship between E and E total as shown, where I is the variable and L and K sub I are parameters. Now I look at the rate law itself, which is a rate constant times the concentration of S and E. And now I can take the equation on the former slide and substitute it in there. Here I have put a general number of subunits gamma into the expression, and on the previous slide gamma was 4, but you can actually derive this for any number of subunits of a regulated enzyme. This gives me the rate law that I'm after. I get the reaction rate as a function of the substrate concentration and the inhibitor concentration. And there are a number of kinetic parameters in it, in addition to the total enzyme concentration. There is a similar set of equations I can derive for serial binding of activators to the relaxed form, the squared icons in the former slide that I had, and show how an activator pulls more and more of the enzyme into the active form. And some versions of the symmetry rate law have also substrate loading taking place. The derivation of such rate laws follows the exact same procedure as I've been going through, but with increasingly cumbersome mathematics. Now, on to simulation. I can do the same thing for the symmetry model in a closed setting as I did for the Hill rate law, and the results look very similar. Here I am simulating a condition where I initially have substrate and free enzyme starting at time 0 in the upper right-hand corner of the first phase portrait or phase plane. Phase portrait and phase planes are synonymous here. I see an immediate vertical drop in this phase plane where the inhibitor is loading the enzyme up, and once those dynamics have unfolded, the reaction itself takes place, and I see a horizontal motion where the substrate is fully consumed. There are two other phase planes in here that represent the total amount of enzyme and the total amount of inhibitor. And these are the minus 45 degree type lines, although in this case they aren't exactly minus 45 degrees in all cases. Although I guess the way I've formed the expressions here, they actually are. They go from 1,0 to 0,1. We can also look at the dynamic responses and plot the concentrations as a function of time, and that's what's shown here. The fast response for these parameter values given show the rapid loading of the enzyme with the inhibitor, the decay of the free form, and the buildup of the summation of the inhibited forms. And that is shown here with very little product being made, little substrate being consumed. Then if you look at the longer time scale, a few time units, we see that the substrate is being depleted while the product builds up, and the different forms of the enzyme are steady. All right, now, as I indicated, there are many rate laws that can be developed, and if you're interested, you can look at this book by Siegel. That is about 3 times the thickness of the book that comes with this class, as shown in the photograph, and the little cartoon, that little joke below shows the feeling many express when they start reading through a book like this, and that is that the derivation seemingly go on forever, and you start to understand the concept of infinity as you go through mechanism after mechanism after mechanism and derive these equations. For the purposes of this class, knowing the Michaelis-Menten mechanism, the Hill mechanism, and this simple form of the symmetry model is sufficient. So let's summarize what we have done in here. First, enzymes are highly specialized catalysts that accelerate reaction rates. Normally, they have reaction specificity and substrate specificity. Sometimes the substrate specificity is low, leading to what's called a promiscuous enzyme. It can act on many different substrates. To describe the rate law, we have to formulate first the reaction mechanism, namely the underlying reactions that describe how the enzyme functions. And these are described in terms of the actual reactions that take place or the elementary reactions. The rate laws for the enzymes are then derived going through a systematic process, and historically this includes the application of simplifying kinetic assumptions, mostly the quasi-steady state and the quasi-equilibrium assumption. In this day and age, you can simply assess the validity of these assumptions by simulating the model before the assumptions are applied and compare it to simulation of the simplified model. You can also get at the validity of these assumptions, scaling the equations and going through an advanced analysis procedure that I'm not going to describe here. A number of rate laws have been developed for different enzymes, both regulated and non-regulated. We only described 3 of these reaction mechanisms in here. If you need to treat others, you need to look up information in books like the book I pictured by Lee Siegel and consult the appropriate chapter in there and extract the rate law that is applicable for the enzyme that you're interested in. So that concludes this lecture on enzyme kinetics, and it summarizes what you need to know about enzymes to successfully build the simulators that we will cover in this class. Thank you for your attention.